Danielle and Marlene go and see a plant biologist called Dr. Jennifer Neale. They show her the rare flower and she takes it in to study it. She has asked Marlene and Danielle to help her discover what this plant is.
Okay, let's look at this under the microscope and see if we can see anything else about it that might help us figure out what it is. So this is our microscope that allows us to look at things really close up. So let's see what we can see. That's really unusual. I've never seen anything like this before. This is our lab assistant, Anna. She's going to help us today with some of our DNA PCR and then running the gel so that we can look at this flower and see if we can figure out what it is. So let's see. We can look at it up close here on the computer screen. I don't know. I still don't really know what it is. I think we're going to need to extract some DNA to figure it out. All right, let's go to my office and see if we can figure out what you guys brought in. All right, so if you guys want to come back to my office. So come on into my office and we can sit down and look at some of our rare plants. So this is actually a book of Colorado rare plants, so we can look in here and see if your plant is in any of these. What do you guys think? Do any of these look familiar? Gosh.
I was wondering if you could tell us more about what you do.
And why did I decide to be a biologist? It's kind of a hard thing. I can't say that I grew up knowing I wanted to be a biologist, but because I spent so much time outdoors growing up, I think I just was always attracted to studying the outdoors and learning biology. And so when I went to college, I was a biology major, and it was during that first year of graduate school that I took a class where I learned about conservation genetics, and I learned more about how you can use DNA tools to help conserve plants. And I thought, that's what I want to do. Through our conservation work we do here at the gardens, we collect seed. And then when we collect seed, we want to know how many of those seed are able to make new plants, or how many or what percentage of those are considered to be viable. When we're doing DNA extractions, we add some of this, take out some of that, and so you can add in the liquid and take the liquid out. So we use pipetters, which I'll show you guys. In order to do the DNA extraction, we have to take that leaf and we have to break it up to begin with. So first we break it up before we then add the chemicals to help break it up. And we break it up in a machine called the bead beater, where basically it's like a tiny paint shaker. We put the tissue in a tube with some glass beads, and then we turn it on and it shakes it up like this, and basically just turns that tissue or that leaf into powder or into little tiny pieces, and then we add the chemicals to help break it up. And using pipetters, we add the correct volume of each of these different liquids into one what we call reaction. So it's essentially following a recipe. And then we put that into the thermocycler, which will heat up and cool down that DNA to make millions and millions of artificial or fake copies of it. And then after you run your samples through the thermocycler and you make those millions of copies of DNA, you want to look at them. And so you can't really look at them when they're all bunched together in a tube. You still can't see them, it's just clear liquid. So then we put it on a UV light table, which is essentially a black light, and we take a picture of it that way. And the UV light table makes the DNA glow. And then we can take a picture using the camera hooked to our computer. And this is an example of what that looks like. So each one of these columns is a different individual, and each one of these little glowing bands is DNA. And the reason it's glowing is because we added that certain chemical to it that binds to the DNA and makes it glow when we put it under the UV light. And so this is what the data look like. So we essentially go from having leaf tissue to having a column of glowing bands. Where did it go?
Nothing.
Where Where do you guys think it went?
No.
I've never had this happen before.
Oh my
Gosh, I don't understand. Where do you think it went?
I don't know. It can't hide in there. This is messed up. Let's split up and look for it.
All right.
Some type of mutated giant plant that's hungry. There it is! Ah! Oh my gosh.
Girls!
Let's go! Let's go before it attacks more people! Let's go! Run!
It is so humid in here. It could just grow so big. We have no idea where it could be. Do you guys see it anywhere? There it is! It's changed!
It's talking.